Hi everyone, I hope everyone's having a lovely day. So for today's class, we're going to be learning about genetic information and protein synthesis. So first, we're going to be discussing DNA structure and replication. Okay, let's begin discussing DNA structure and DNA. So DNA stands for deoxyribonucleic acid, which stores master copy of genetic code. It also carries the genetic instructions used in growth, development, functioning, and reproduction. Structure of DNA is a double helix made out of nucleotides. Each nucleotide consists of a phosphate group, a sugar, and a nitrogenous base, which is adenine, thymine, cytosine, or guanine. When it comes to the base pairing rule, adenine pairs with thymine and cytosine pairs with guanine. Okay, so let's dive into this picture of what DNA looks like. So the image of a nucleotide is right here, this image I circled. So a nucleotide is the basic building blocks of DNA. This diagram shows the three components of a nucleotide. There is a phosphate, which is right here. Then we have our sugar, and then we have our nitrogenous base. This image also shows base pairing rules right here. This is our base pairing rules, which explain how the four nitrogenous bases pair up in DNA. The image to our right is a diagram of a double helix, which is also a three-dimensional structure of DNA. So this is a double helix. The double helix is formed by two chains of nucleotides that are linked together by hydrogen bonds between the base pairs. Okay, let's talk about process of replication. So this occurs during the S phase of the cell cycle. So when cells divide, each daughter cell must receive identical DNA. So when it comes to the steps in DNA replication, we first have hydrogen bonds break between base pairs. So this means the double helix structure of DNA is held together by hydrogen bonds between the base pairs. These bonds break, separating the two DNA strands. Next, we have strands unwind and separate. The two strands of DNA unwind and separate, creating two template strands. Third, new nucleotides pair with exposed bases under direction of DNA polymerase. So this means new nucleotides, which are building blocks of DNA, pair with the exposed bases of the template strands. This pairing is guided by the enzyme DNA polymerase, which ensures that the new strand is a complementary copy of the template strand. Lastly, we have other enzymes connect new sugar phosphate backbone. So other enzymes connect to the new nucleotides together to form a new sugar phosphate backbone, creating a new DNA strand. Okay, so let's break down DNA replication with this image we have right here. The left image that I circled shows a double helix of DNA, which is the original DNA molecule right here. In this image, there's also two new DNA molecules that are being formed. 1, 2. The process of DNA replication is shown in this image as the original DNA molecule is being unwrapped and copied. The DNA molecules are also being shown being separated and forming two new cells. The image up here shows the different phases of the cell cycle, which are G1, S, G2, and M. If you recall from my last slide, DNA replication happens in this S phase right here. Okay, so now let's dive into some protein synthesis. Protein synthesis is the process by which cells create proteins which are essential for various functions in the body. It involves two main stages, transcription and translation. During transcription, the DNA sequence of genes is copied into messenger RNA, which is mRNA. mRNA is a type of RNA that carries genetic code from DNA to ribosomes and cytoplasm. The enzyme RNA polymerase binds the DNA at the start of the gene and unwinds the DNA strands. Then RNA polymerase is an enzyme that catalyzes the formation of mRNA from the proper strand of DNA. Here we're going to discuss the steps of transcription. So we start with RNA polymerase right here. RNA polymerase recognizes the correct strand of DNA to copy. So this is the strand of DNA. So RNA polymerase binds to promoter, which is right here. and then the DNA sequence that signals beginning of a gene. Then a section of DNA unwinds to expose the gene coding for the particular protein. So this is the sequence of DNA that's unwinding to expose the gene coding. Then we have a complementary mRNA nucleotides pair with the DNA bases. Then termination signals indicates end of gene, and neuromRNA strands is released. Here. And then DNA rewinds into a double helix, as you can see. It's happening right here. The mRNA now leaves the nucleus through a cellular pore and attaches to a ribosome in the cytoplasm. And this is how transcription occurs. Now we talk about translation. So this takes place in the cytoplasm where ribosomes read the mRNA sequence. Translation is a process of converting the genetic code carried by mRNA into a sequence of amino acids that become a protein. Okay, so the process of translation consists of 4 steps: initiation, elongation, translocation, and termination. So in initiation, the ribosome binds to the mRNA at the start codon. Then in elongation, the ribosome moves along the mRNA, reading the codons and adding amino acids to the growing polypeptide chain. Then in translocation, the ribosome shifts one codon down the mRNA. And then finally in termination, the process continues until a stop codon is reached and the completed polypeptide chain is released. Here I have added a picture walking us through the steps of translation. Translation is right here, and then right here we have our polypeptide chain right here, of course, and this is translation, and then this is transcription right here. Throughout this presentation, I have picked up on some of my important questions I wanted to ask you guys. Here is how I would answer the questions I have asked you guys. You guys can match them up, see them. If there's any questions, you guys can ask me. But thank you for listening to my presentation.